Welcome to Wildcat Science with Mrs. Haugo. This is Human Biology, Chapter 3, Part 2, and we are talking about cells and their functions. And we talked about what's wrong with Jim. Previously in Part 1, we talked about the 3 different types of microscopes and how they help look at cells. We talked about the plasma membrane, the nucleus, the components of the plasma membrane, the bilayer, the cholesterol, carbohydrates, and proteins, the different proteins in the plasma membrane. We talked about membrane potential, how that's a difference in electrical voltage on either side of the membrane. It's used to do work like generate ATP, or it's used in communication like in an action potential in nerves. Then the nucleus is also very important. It contains the DNA, controls everything that happens. It's involved in protein synthesis and mitosis. And then cytoplasm is most of the cell, and that's the organelles like the endoplasmic reticulum and the ribosomes and the mitochondria and the Golgi apparatus and the lysosomes and vesicles and centrioles. And we also talked about different types of cells and how the cell structure determines its function and how that's really intertwined, and did some checkpoints. And then we talked about how that cell membrane is semi-permeable, which means some things can get through but others can't. We talked about osmosis, how that's water moving from high to low concentration, and diffusion, which is other substances moving from high to low concentration. And we talked about facilitated diffusion, also moving from high to low concentration, but it needs a little help. It needs a transporter. And filtration, which is moving under pressure. So now we're going to talk about movement that does require energy. So the first one I want to talk about is active transport. And that is when you're actually moving it backwards. You're going from low concentration to high concentration. And you're using energy to do that. So you're going where there isn't much and pumping it to where there's a bunch. So nature doesn't really want to do that naturally, so you have to put in energy to get it done. Why would we ever want to do this? Well, there's an active transport of calcium ions into your muscle cells so that they're ready to burst out and be used during a muscle contraction. Also, sodium-potassium pumps will pump sodium on one side of the cell membrane and potassium on the other side of the cell membrane so that it's ready to open the floodgates, and then they flow where they want to go naturally, and then that sends a nerve signal down your neuron. So those are 2 very good examples of when we would want to use energy to move things backwards. Okay, so what else does require energy? Bulk transport or vesicular transport does, where you're transporting things in vesicles. One type is endocytosis, and endo would be going in to the cell, moving materials into the cell. And there's a nice picture on page 48 of phagocytosis, where a large particle is like engulfed by the plasma membrane, and then it just kind of pinches off and makes a vesicle, and then it floats to where it needs to go in the cell. So white blood cells use phagocytosis when they are engulfing a virus or a bacteria, and then they digest it and take care of it for us. And then pinocytosis is very similar to phagocytosis, but it's fluid. So it's like cell drinking. The fluid is engulfed by the plasma membrane, it gets into a little vesicle, and it goes where it needs to go. So what is the opposite of endocytosis? Exocytosis. That's moving materials out of the cell. So basically you just do it backwards. There's a nice picture on page 49, figure 313. You have a vesicle, and that molds or blends in with the cell membrane, and then it kind of pushes the stuff out, and the substances go outside of the cell. And there is a nice table on page 49 which lists all the different types of transporting things across the cell membrane. So A review from biology, balancing the amount of water in cells is really important. So isotonic, where the cell, the water content and solute content inside the cell is the same as outside the cell. And isotonic is when they are equal. Hypotonic is when it is low, the concentration of solutes is low, and that's outside the solution, it's low. So the solution has fewer solutes outside than it does inside. And we're going to be looking at a picture, and that's on page 47, which will help you. So what happens if you put the cell in a hypotonic solution? Well, we know water normally goes from high water to low water. So if it's going from low water, it's going to, where there's low water, there's a bunch of solutes. So it'll be going from outside of the cell, where there is not much, to inside of the cell, where there is a lot. So water's going into the cell, and that makes, the water will move from the solution into the cell, and that makes the cell swell and then burst and die, which is bad. We don't want our cells dying. On the flip side, hypertonic, that means higher solution. So the solution outside of the cell is more concentrated, it has more solutes than inside of the cell. And so if you put a cell in a hypertonic solution, we know that water goes from high water to low water, and if it's low water, it means there's lots of solutes, and there's lots of solutes outside of the cell. So the water is going to move from the cell out into the solution, and so then that cell will shrink and die. And again, there's a nice picture on page 45, figure 3.7, and we did this experiment in biology where we had dialysis tubing with 20% solution of sugar, and we put it in a solution of 20%, and then it stayed the same. And then we put it in a solution of 0%, which would be hypotonic, and when that happened, the water went into the cell and it swelled. And then we put it in a solution of 40% water, which was hypertonic, and then when that happened, the water went out of the cell and it shrunk. So in both cases, hypertonic and hypotonic solutions, homeostasis is not occurring and your cells are in danger. So looking at some of our checkpoints, 3.8, what type of movement goes through the plasma membrane does not require energy, and what types of movements do require energy? And 3.9, what term describes a fluid that is the same concentration as the intercellular fluid, and what type of fluid is less concentrated, and what's the term when it's more concentrated? So another thing we talked about was protein synthesis. We talked quite a bit about that in biology, so let's review that. So DNA and RNA are nucleic acids, and you don't need to write this down. If you feel like you've never heard this stuff before, then you probably should write it down. What's a nucleic acid? It is a polymer of nucleotides, and remember, a nucleotide has a sugar, a phosphate, and a nitrogenous base. And there are 4 flavors of nitrogenous bases: adenine, thymine, guanine, cytosine. If you are DNA, and then RNA would have uracil instead of thymine. And if you remember, adenine bonds with thymine. If you're RNA, adenine binds with uracil, if you're RNA. And then guanine bonds with cytosine. And like we said, the difference between DNA, one of the differences between DNA and RNA is that RNA does not have thymine, it has uracil in there. So no T's, only U's. So protein synthesis, please write this down. DNA is the genetic code or the blueprint, and it always stays in the nucleus. And DNA makes up chromosomes. And a section of a chromosome that codes for a protein is called what? If you said a gene, you are correct. And there is a nice picture on page 50 that shows a chromosome and then a section of a chromosome, which is a gene, and that's a little green area. And then they blow that up so you see a section of the double-stranded DNA. And then they blow that up so you just see one little nucleotide that makes up the nucleic acid. And then on page 51, there's also a nice picture of the double-stranded DNA. So protein synthesis, if you remember from biology, it starts with transcription, and that during transcription the DNA unwinds and a complementary RNA single strand is made, and then that RNA leaves the nucleus and goes to the ribosomes, and that's the messenger that brings the information from the nucleus to the ribosomes in the cytoplasm where it needs to be, where that information needs to be. And then that is where the process of translation starts, and during translation, RNA is decoded from a strand of amino acids, a strand of nucleotides, excuse me, into a chain of amino acids. And then once you have that chain of amino acids that have been made from that information, we call that a polypeptide. And you put 1, 2, 3, 4, maybe more polypeptides together, they fold up in this complex shape, and you make a protein. And there is a very nice picture on page 52 that shows a cell and then the nucleus, and then it shows DNA unwinding and making a complementary RNA strand. And then it shows the RNA moving to the ribosomes and an amino acid chain being made. So looking at our checkpoints on page 52, what are the building blocks of nucleic acids? And what category of compounds does DNA code for? And then what are the 3 types of RNA that are active in protein synthesis? And perhaps you'll remember that from biology, or maybe you'll need to look that up in the book. So, cell division for growth, repair, and reproduction. So, sex cells divide by the process of meiosis. You got that right, good job. And that meiosis divides the number of chromosomes in half. So we go from 46 to 23, which is good because this happens in the egg and the sperm. And then when an egg and sperm come together during fertilization, you have the magic 46 number again. And then somatic cells, which is every cell in your body that is not an egg or a sperm, divides by the process of mitosis. If you said mitosis, good job. And in mitosis, you make 2 identical cells which have the same number of chromosomes, which are diploid or haploid cells, if you remember your terms from biology. Okay, the phases of mitosis. If you remember what these are, great, you don't have to write them down. If you have never heard of this before, you should probably write it down. Interphase. During interphase, a cell grows and DNA replicates. Next phase is prophase. The DNA condenses into chromosomes, the nuclear membrane falls apart, the centrioles move to opposite sides of the cell, and then we move into metaphase, where the chromosomes line up at the cell and the spindle fibers attached to the centromeres in the middle. And then we have anaphase, where these duplicated chromosomes separate and they move to opposite sides. And then we have telophase, where that nuclear membrane reforms and the plasma membrane starts to pinch and make 2 identical cells. If you remember doing the pop beads and going through all the steps of mitosis, that is what we did. Hopefully you can picture those and remember that in your brain. Okay, so mitosis checkpoints. 313, what must happen to the DNA in a cell before mitosis can occur, and during what stage in the cell's life cycle does this thing occur? And 314, what are the 4 stages of mitosis? So let's talk about cell aging, and I think we're going to do that in a short 3rd video, and I will see you then. Bye.